Ladies and gentlemen, good morning. So before anything else, I would like to enumerate the subtopics that will be discussed later. So first, the introduction of gene therapy. Second, the basic process of gene therapy. Third, the two types of gene therapy. Fourth, the differences and similarities of the two types of gene therapy. And lastly, the significance or purpose of gene therapy to humans. So my task here is to introduce to you what is gene therapy. So before I start, I would like to introduce myself. So I am Angela Oberes from BSIT2 Black 2, and it's my pleasure to discuss with you what is gene therapy.
Genes make us who we are. Are we tall or short? Do we have blue eyes or brown? Our genes hold the answer. What if part of a gene is defective or missing? Or if a gene changes or mutates? It can cause a disease like cancer. Enter gene therapy. Among other things, scientists can replace a bad gene with one that is healthy, add a new gene to perform the function of a defective or missing gene, or they can turn off a problem gene. How do they do it? Say scientists want to add back a healthy copy of a gene to cells. They need an efficient way to do it. So scientists use vectors or vehicles to deliver a gene. The healthy gene hitches a ride in the vector. Viruses are one kind of vector. Scientists modify a virus to remove its ability to cause a disease. The modified virus can then be used to carry the healthy gene into a cell. Gene therapy can happen on cells inside the body, or can happen on cells outside, and then transplanted back in again. Genes make us who we are, and gene therapy holds great promise for a healthy future.
So what is gene therapy? So gene therapy is defined as the treatment of disorder or disease by transfer of genetic materials into cells. So it is an intracellular delivery of genes to generate a therapeutic effect by correcting an existing abnormality. So gene therapy replaces a mutated gene with a healthy ones inside your body. So it also introduces a new gene in the body to help fight the disease, and it enhances the effect of a normally functioning gene. So gene therapy can be used to modify cells inside or outside the body. So gene therapy was invented by W. French Anderson with his colleagues at NIH on September 14, 1990, who performed the first approved gene therapy procedure on a 4-year-old girl born with severe combined immunodeficiency. So French Anderson was also the father of gene therapy. So the concept of gene therapy arose initially during the 1960s and early 1970s. So the first gene therapy was licensed in China on 2003. Gene therapy holds promises for treating a wide range of disease such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. So in United States, gene therapy is available only as part of a clinical trial. So that would be all. So the next video will help us to understand more about gene therapy, and also it will give us more additional information about what I have discussed earlier. So please fasten in your seatbelt and listen carefully.
Hi everyone, my name is Jessa Marie Menigopolo, and I'm going to discuss with you the basic process of gene therapy. But before that, let's do a recap to the previous discussion. What is the gene therapy? So, gene therapy is defined as the treatment of disorders or diseases by transferring the genetic materials into the cells. It is intracellular delivery of genes to generate therapeutic effects by correcting an existing abnormality. So now we're going to mention or discuss and explain the steps of process of the basic gene therapy. So the step 1 is cells harvested from the patients. Usually, the cells are removed from the patient's own bone marrow, as well as the stem cells can be harvested from the blood or the bone marrow as well. The step 2, virus altered or reformed so it cannot reproduce. Cells takes place in the lab where the gene must not replicate itself. Step 3, a gene is inserted into the virus. So a gene that is inserted directly into the cell usually does not function. Instead, a carrier called vector is genetically engineered to deliver the gene. So certain viruses are often used as vectors because they can deliver the new gene by injecting the cells. The step 4, altered virus mix with the patient cells. So in order to insert the new gene directly, they use a vector which generally engineered to deliver the gene, as I mentioned earlier. So step 5, cells become genetically altered. So gene therapy seeks to alter genes to correct genetic defects and thus prevent a cure genetic diseases. Step 6, altered cells injected into patient's body. So gene therapy involves altering the genes inside your body cells in an effort to treat or stop diseases. Genes contain your DNA, the code that controls much of your body's form and functions. Step 7, altered cells produce desired protein. So this is now the cells continues to multiply and eventually produce the desired effect. So now let's watch this short video about how gene therapy works, though it does not mention the basic process, but it will help you to understand the flow of the process.
Gene therapy is a treatment strategy that addresses a disease condition by introducing new genes into cells. In contrast to drug treatments or surgery, this technique is designed to address the root cause of the pathology. The process often begins following a positive diagnosis for a disorder approved for treatment by gene therapy. Viral vectors are commonly used in gene therapy to safely transport a therapeutic gene to the target cells. To ensure high efficacy and patient safety, rigorous vector purification and characterization is crucial. Scientists grow cells which produce gene therapy vectors in a bioreactor. During the process, cell growth and viability is monitored, along with the surrounding media and metabolites. Maintaining optimal, consistent growth parameters can maximize production and help mitigate resource waste. Once the cells have adequately grown, the cells are quickly and reliably separated from cell culture media with a high-performance centrifuge. Viral vectors may be secreted into the media or retained in the cells. In this scenario, we will focus on the cells. Cells within the pellet are resuspended and then lysed through mechanical or chemical methods, which releases the gene therapy vector. For low shear and minimal product loss, cellular debris can be removed through high-performance centrifugation, where biosafe systems ensure operator and sample protection. Next, particle separation through density gradient ultracentrifugation provides high yields of capsids with high purity. For both step and continuous gradients, the desired viral vector and contaminants can be found at discrete locations corresponding to their respective densities. Typically, full capsids are desired, and these more dense vectors are found lower in the gradient. The band of interest may then be extracted. A small amount can then be analyzed for quality control and characterization, while the majority of the liquid is packaged into vials ready for shipment. Quality control is crucial throughout gene therapy production. High-resolution native state analysis with analytical ultracentrifugation confirms that the finished product consistently contains mostly full capsids. AUC is the industry standard technique to quantify packaging efficiency of viral vectors. After passing the QC inspection, the gene therapy is shipped to be administered to the patient.
So, in general, a gene cannot be directly inserted into the person's cells. It must be delivered to the cell using a carrier or vector. So, vectors system can be divided into two, which is the viral vectors and the non-viral vectors. So, what is vector delivery? So, vector delivery in molecular biology, a vector is a vehicle that used to deliver genetic materials to target cells via horizontal gene transfer. So there are several methods for transferring genetic material, but all mechanism can be classed as either viral or non-viral. So the first vector delivery is the vector delivery. So the insertion of a viral vector into a cell is called transduction and utilize the ability of the virus to inject its DNA into the host cell. So viral vectors are typically used due to their ability to integrate DNA into the host genome. So the second one is the non-viral delivery. So a commonly used vector is a plasmid, a circular DNA molecule capable of self-replication and autonomous protein expression. So viruses can have either a DNA genome or the adenovirus or an RNA genome or the retrovirus. Currently, the most common type of vectors are the viruses that have been genetically altered to carry normal human viruses. So the virus have evolved a way to encapsulating and delivering their genes to the human cells in a pathogenic manner. So scientists have tried have tried to harness the ability by manipulating the viral genome to remove diseases causing genes and insertion of therapeutic ones. So target cells such as the patient's liver or lung cells are infected with the vector. So the vector then unloads its genetic materials containing the therapeutic human gene into the target cells. The generation of the functional protein product from the therapeutic gene restores the target cells to a normal state. So as we go on to the discussion to the gene therapy, we all know that gene therapy is so amazing and can treat many diseases. But have you ever wondered, is there a person who died or got injured due to the gene therapy? Unfortunately, guys, there is who have volunteered to to a clinical trial. His name is Jesse Gilsinger. He died on September 17, 1999. He was the first person publicly identified as having died in a clinical trial for gene therapy. The viral vector caused a fatal immune system. So the 17-year-old Jesse Gilsinger had a rare metabolic disorder called ornithine transcarbamylase deficiency syndrome, or OTCD, in which ammonia builds up to the lethal levels in the blood. So the previous patients in the trial had experienced flu-like symptoms, but had much worsened reaction within a day. So within a day, he became disoriented and showed sign of jaundice. So he had an intense inflammatory response and developed a dangerous blood clotting disorder, followed by kidney, liver, and lungs failure. For 4 days after receiving the shot, Jesse was declared brain dead and taken off life support. So the team of doctors and nurses caring for him were stunned by his rapid decline and death. So the news that an experimental treatment had killed basically healthy volunteer rocked the field of gene therapy and the broader world of biology research. So due to the case of Jesse Gilsinger, the risks should be visible as this experimental is continually developing. So the gene transfer that uses an adeno-associated virus AAV vector to deliver the new genetic material may have several risks. So as with any virus, the body's immune system could respond to the newly introduced therapeutic vectors as it is were an intruder. So an immune system reaction can lead into inflammation and other serious risks. So an immune reaction could make gene therapy work less effectively or not at all. So that is why prospective gene therapy patients are often screened using a simple blood test and determine whether they have antibodies against a specific virus. So while the objective of using a particular vector it is to direct to the new gene to the specific tissue type. So viruses can affect other cells that weren't targeted, so potentially causing damage or additional illness or diseases. After delivering of the gene therapy, vector particles can be cleared from the recipient's body. It's called vector shedding. This can occur through feces, urine, saliva, and other excreted bodily fluids. So shedding raises the possibility of passing those remaining materials to untreated individuals through close contact. So ongoing gene therapy studies are currently evaluating the significance of vector shedding. So whether gene therapy may have an adverse impact on the health of the organ or tissues target is being evaluated with long-term study. Gene therapy may result too much of the protein being created. The effect of this overproduction or expression could vary based on the type of protein being created. As with all medication, response to gene therapy may vary across patients. Though hundreds of volunteered patients who experience the clinical trial get injured or died, there is also a bright side in gene therapy. An example of the successful use of gene therapy in the human. So in 1990, a 4-year-old Ashanti Di Silva became the first gene therapy success story. He was born with a severe combined immunodeficiency. due to the lack of enzyme adenosine deaminase. Without ADA, her T cells died off, leaving her unable to fight infections. Infections of synthetic ADA enzyme help, but only temporarily. Doctors decided to deliver a healthy ADA gene into her blood cells using a disabled virus that cannot spread in the body. So their success spurred more trials in the 1990s for the same form of SCID. Now, in her 30s, Dee Silva is active in the rare disease community. That's all for me. Thank you. And next topic would be the two forms of gene therapy.
Good day, everyone. The first type or forms of gene therapy was the somatic gene therapy. So, what is somatic gene therapy? Somatic gene therapy is the transfer of genes into the somatic cells of the patient. It is the cells that does not produce the eggs and sperm, in which it produces the next generation, such as the cells of the bone marrow, and hence the new DNA does not enter the sperm or eggs. The genes transferred are usually normal alleles that could correct the mutant or the diseased alleles of the patient. The strategy of somatic gene therapy is persuasively very simple. The disease is caused by abnormal gene or DNA expression, so the treatment of the cause should be possible by correcting this abnormal expression using DNA or, more generally, nucleic acids. Somatic cell gene therapy would aim to cure a disease only in the patient, but not in the patient's descendants. Somatic gene therapy has two types, as shown in the diagram above. The two types of cell gene therapy are possible: the ex vivo and the in vivo. So the ex vivo form, the normal gene is introduced to cells removed from the patient. Then the treated cells are returned to the patients via the circulation. While in vivo therapy differs in that the normal gene is introduced directly into the patient, generally into the tissue requiring the treatment, such as muscles, liver, or brain. Both types, the normal gene must be incorporated into a vector, or usually a non-pathogenic virus, and propagated to obtain a sufficient number of new gene copies to make treatment clinically feasible. Immunodeficiencies, hemophilia, thalassemia, muscular dystrophies, and cystic fibrosis are some example or common types of disease which could be potentially treated by somatic gene therapy. There are some examples of somatic gene therapy have already proven to be clinically effective. The first successful treatments of adenosine deaminase deficiency took place in 1990 in 1991 with two patients aged of 4 and 11. Both are thriving with continuing treatment. Then the first successful treatment of familial hypercholesterolemia, a genetic condition which affects the liver's regulation of cholesterols in the blood. It is took place in 1992 of a 29-year-old woman. Her improvement was stable for the 18 months of the study, and liver biopsy demonstrated activity of the inserted gene and no discernible abnormalities. 5 patients have been treated as of 1994. Current research involving somatic gene therapy is focusing on a number of areas. Clinical trials are being performed on a treatment for cystic fibrosis, a chronic genetic disorder.
The second form of gene therapy is germline gene therapy. Germline gene therapy is when DNA is transferred into the cells that produce reproductive cells, eggs or sperm in the body. This type of therapy allows for the correction of disease causing gene variants that are certain to be passed down from generation to generation. However, for many, this is where they feel the benefits of germline gene therapy stop. Germline gene therapy is currently not legal in the UK because the risks still appear to outweigh the benefits. Germline gene therapy. In germline gene therapy, DNA is inserted into the reproductive cells, eggs or sperm in the human body. Germline gene therapy will correct the genetic variants of the reproductive cells of an individual, and this will be passed down to future generation. This therapy removes a hereditary disorder from a family line forever. Hereditary disorders occurs as humans are possible inherited from the germline cells. However, curing this disease is possible only through modifying their nuclear and mitochondrial DNA mutation in fermentation embryos, which is commonly known as germline gene therapy. This picture is the virus vector of germline gene therapy. The virus vector should be engineered so that it only introduces the DNA to a particular type of cells in the body. This can be very difficult and is one challenge scientists are trying to overcome to use gene therapy. An alternative is to remove the cells from the body and transfer the DNA in a lab, then return them to the patient.
Hi everyone, my name is Almera City Mario BS82 Block 2, and now I'm going to report about the differences and similarities of the two forms of gene therapy, which is the somatic gene therapy and germline gene therapy. There are important differences between the two. Somatic therapies target genes in specific types of cells: lung cells, skin cells, blood cells, and etc. While germline modifications applied to embryos, sperm, or eggs alter the genes in all the resultant person's cells. Somatic cell modifications are non-inheritable, affecting only the treated individual. Germline modifications will be passed on to future generations. Somatic cell therapy have been tested and implemented for much longer. The first somatic trials occurred 2.5 decades ago, while human germline editing studies have only just begun this year. Similarities: somatic and germline gene therapies involve altering of defecting genes or introducing of healthy genes. Both therapies use genes transformations method.
Hi, my name is Ryan Pakatang from this 82 Block 2. So today I'm gonna report about the significance and purposes to gene therapy to human. So gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight diseases. Gene therapy holds promise from treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Gene therapy is used to correct defective genes in order to cure a disease or help your body better fight disease by replacing mutated genes. Some cells become diseased because certain genes work incorrectly or no longer work at all. Replacing the defective genes may help treat certain diseases. For instance, a gene called P53 normally prevents tumor growth. Several types of cancer have been linked to problems with the P53 gene. If doctors could replace the defective P33 gene, that might trigger the cancer cells to die. Fixing mutated genes. Mutated genes that cause disease could be turned off so that they no longer promote disease. Or healthy genes that help prevent disease could be turned on so that they could inhibit the disease, making disease cells more evident to the immune system. In some cases, your immune system doesn't attack disease cells because it doesn't recognize them as intruders. Doctors could use gene therapy to train your immune system to recognize the cells that are a threat. So the advantages of this technique to human is to give someone that is born with a genetic disease or who develops cancer the chance at a normal life. I think this is an advantage that for our ways any of the advantages that have been presented against gene therapy. Giving someone a chance at a normal life should be enough for many of the people that oppose this technique to change their minds. I believe the reasons that these people are opposed to gene therapy is because they see it as something foreign and scary. The reason people are scared is because they do not understand the technique. I think if scientists educate our society, many people would change their minds. I believe that if the people that oppose this technique or were ever faced with cancer or a child born with a genetic disease, they would change their views. These skeptics would choose gene therapy, especially if it would save a loved one's life. In the coming future, gene therapy will play an important part in many people's lives. So, how many people that are successfully treated and successfully done in this gene therapy technique. Since 2016, more than 10,000 people have been treated with it worldwide. Although Spinraza can't restore completely normal motor function, a single motor neuron gene just can't produce enough protein for that. It can help children with any of the 4 types of SMA live longer and more active lives. So that would be all. Thank you.